So, have you ever worked with microsatellites before?
No, never. So I'm nervous, but I'm excited.
That's literally it.
My name's Claire Mauss, and for the next 10 weeks, I'm going to be trying to do research with microsatellites.
Anything else you need to say today? Actually,
Wait, I can keep that in, that's
fine.
Looking at genetic data using microsatellites is a useful way of tracking recent changes to populations. Microsatellites rely on neutral genetic diversity, targeting non-coding regions of the genome, which exhibit higher mutation rates than coding regions since they are not subject to the same pressures of selection. I'll be using 6 microsatellite primers in my research that are designed for hibiscus species. Currently, there are 5 known endemic Hawaiian hibiscus species, which include 9 taxa that share a single ancestor from one of the 4 independent colonization events of the genus. Of these 9 taxa, I will be looking at Hibiscus waimeae subspecies waimeae, Hibiscus waimeae subspecies Hannerae, and Hibiscus clayi. The last of these 2 are internationally listed as endangered, which is why it's so vital that we study these species. So the first step of this process is going to be extracting the DNA from my Hibiscus clayi samples. I have tissue from almost every single individual that exists on the islands of Hawaii, from 2 different populations. So I have about 100 samples, so these extractions are going to take a while. The standard DNA extraction procedure for plants is done using a method called CTAP. But since hibiscus are in the Malabaceae family, they have a bunch of mucilage, so they have a modified procedure that takes 2 days instead of 1, so I have a lot of work ahead of me. So my next step is to amplify the DNA that I just extracted using a polymerase chain reaction and my primers. I'm wanting to use 6 primers, so fingers crossed that they all work.
So how's it looking?
So these are my first runs, and they're looking great. Most of the data showed up, so I'm really excited. But nothing gold can stay. Quickly, I started running into problems with 2 of my primers, HWA71 and HA13. So some of my primers aren't working. They're just showing up as noise. So I'm not really sure what to do next, and with so little time left, I might just have to scrap them altogether. I tried tons of different PCR recipes to try to fix the problem, using different DNA concentrations, varying the primer concentrations, trying different annealing temperatures, but all to no avail. With so little time, I had to make the difficult decision to scrap those 2 primers and only move forward with the ones that were working. Despite some complications along the way, I was able to collect most of the data from my other 4 primers. From this data, I found that the populations of the endangered species Hibiscus clayi and Hibiscus waimeae subspecies Hannerae exhibited less diversity than the non-endangered species Hibiscus waimeae subspecies waimeae. The 2 endangered species had lower heterozygosity levels and greater inbreeding levels, which could be potential causes of their endangerment. Additionally, I found that larger populations displayed greater inbreeding levels than smaller populations, which was surprising. Inbreeding depression in smaller populations could be a potential reason why this occurred. If inbreeding depression was occurring, it would mean that there was so much inbreeding in these small populations that the overly bred individuals became so unfit for their environments that they died off very quickly, and therefore were not represented in the data. My time at the Chicago Botanic Gardens taught me a lot about the trials and errors of the scientific process, and strengthened my desire to go to graduate school and continue studying plant biology. I'm so grateful to have made so many incredible friends along the way and have had such a supportive group of mentors around me during these 10 weeks. I will forever be appreciative of this summer and bring the skills and memories with me wherever I go in the future.